Welcome. It is so great to be here tonight. I am really happy to have you here with me. This is Mr. W, and we're doing a special review session because we know that semester finals are coming up, and we are really excited. I'm really excited to help you do some review. So it's fantastic to have you here on the um, review session that we're about to do. Want to let you know that this is part one of two sessions. The second one is going to be on Thursday night. That's December 5th. And I'm really happy to see so many people here. And in fact, what would be great, it would be if some people actually just chatted in and let me know two things. One is, can you hear me? I'm always curious about my audio and how the audio is working. So something like sounds good, Mr. W would be a fantastic thing to hear from some of you. And the second thing is, tell me where you're from, what school you go to, more importantly, what state you're in, and when your final is. So I'd love to know that, and just chat in and let me. And I want to thank Gabriella because she told me that I sound good. And that is a good feeling because I've had some traumatic experiences on YouTube where I've talked for a long time. Nobody heard me because my audio wasn't working. But that's in the past. Now we're moving forward to better and funner times. And let's talk about what's going to happen tonight. So tonight's focus is on units 1 and 2 of AP Bio. And again, the goal is to get you ready for your AP Bio Semester 1 final, which is probably happening sometime in the next 2 or 3 weeks. Session 2, again, is on Thursday, 12/5. That's going to be at the same time. We're going to start at 6 p.m. PST, and we'll do units 3 and 4 on Thursday night. So I'm going to tell you about some resources for Semester 1 review to start. Then we're going to talk about your questions. That's really the main purpose of tonight's show. I have a slide deck that covers all of units 1 through 4, but my goal is not to go through that slide deck. My goal is to respond to your questions. So I hope that you're ready, and that's what we're going to do. And I see a question from Ella, and she's asking if the lives stay on the channel after the stream. Yeah, they stay there. You can see lives that I did last year for AP Bio review for last year's AP Bio exam. So yeah, the lives just stay there. Sometimes what I do is I download them, edit them, and clean them up. And then basically you have two ways to access the same material. But the material is going to stay there. So you can watch again and again. You can share it with your friends. You know, the beauty of YouTube. And if we have time, we'll do a few practice FRQs. The thing about FRQs for tonight's content is that there are not that many FRQs that are really related to just units 1 and 2, and that's our focus for tonight. So if you come back on Thursday night, that's when it'll be a little bit easier to answer your questions and do those FRQs. So that having been said, let me tell you about a couple of resources. Resource number 1 that I want to tell you about is that on Saturday morning, it'll be morning here on the West Coast, it'll be afternoon in other places, but that's on 12/7 at 10 a.m. Pacific Standard Time. It's 1 p.m. Eastern Time. I'm going to hold another session like this, but it's going to be on Zoom. And it's not going to be free. You'll have to pay for it to get admission. And my main focus is going to be answering questions, reviewing content, talking about FRQs, and talking about study tips. And if you want to sign up for that, just go to apbiosuccess.com/sem1review. You can learn about it, sign up, you can pay, and that would be awesome. So that's one thing that you can do to get yourself in the best possible position for succeeding on your semester 1 final. And again, my goal and your goal are completely aligned. I want you to get an A. You want to get an A. We're in this together. Second thing is that there's a ton of practice that you can do in the form of flashcards, multiple choice questions, practice FRQs, games that are available for you on my website, learn-biology.com. It's a complete AP biology curriculum that's been approved by the College Board. It's being used by thousands of students, hundreds of teachers around the world, and you should use it too. So go to learn-biology.com, find out about subscribing, and it's going to be your ticket towards having massive success in your AP Biology course this year. And here's the third thing. You need some kind of checklist to do effective studying, and I've put one together for you. And so you can go to apbiosuccess.com/checklist, download my checklist. It lists all the topics for AP Bio 1 through 8 for the whole course, really. So you can use it throughout for your reviewing for the semester final and then for the rest of the year. But it's a good idea to have a checklist. Now I've made one for you. Download it. It's only going to help you in your effort to crush it on your upcoming semester final and in the rest of your AP Bio class. So now things need to get very interactive, or it's going to be a very boring show. And I'm really grateful to people like Gabriella and and Tishal and Ella who have already chatted in. But now I want some other people's opinions, because I want to know what your questions are, because your questions are going to guide what I'm going to do. There's too much stuff for me to just go in a linear way through all the topics that you've covered so far since you started learning AP Biology, either in August or September, depending on where you are in the country. So what are your questions? Go ahead and look over this list, and whoever chats in first with a question, that's what I'm going to start teaching about and reviewing about. And, make your questions as specific as possible, and I'll be able to give the most specific answers. And again, this list is limited to content that's from units 1 and 2. That's biochemistry and cell structure and function. We're going to do units 3 and 4 on Thursday night. So as much as you might want to review stuff like photosynthesis and the Calvin cycle and stuff like that, let's do that on Thursday night. I'm seeing a typo here, and I can't stand it when that happens. So I'm just going to go like this. I'm going to go here. I'll go here. So much better. Okay. So I see a question from Gabriella. Osmosis and water potential relationships are confusing to me. Gabriella, that's fantastic. That's exactly what I want to hear. So let's see. The best way to do it is for me to go like this, and I'm going to go like this. And I've just got so much stuff here, and that's all the way at the end. Osmosis, membrane transport, tonicity, and osmoregulation. Okay, let's go. Okay, so tonicity and osmoregulation. That is what the College Board calls this. So let's go through it. Osmosis, it's just the diffusion of water. So I've got a setup over here, and let me get my laser pointer. Here we are. So I've got a setup over here. Basically, what we've done is we've poured some solute into the left side of this container, and this container is divided by a selectively permeable membrane. If you are an AP Bio student, which I'm assuming most of you are, then you did a lab that was essentially like this, and the semi-permeable membrane that you used was dialysis tubing. So basically, what happens is that water flows down its concentration gradient, just like everything else. It flows from higher concentration to lower concentration. And that means after some period of time has gone by, water will move from here to here. The water level on the left side will rise. The water level on the right side will fall. And that's assuming that this selectively permeable membrane is permeable to water, but not to the solute. And again, in a typical AP Bio lab, the solute would be something like sucrose, and water is water. So that is what osmosis is. Water flows down its concentration gradient from hypotonic to hypertonic. And here we'll define what these terms are. Hypotonic means relatively more water, relatively less solute. This side is hypotonic, relatively more water, like maybe 98% water and 2% solute. This side is hypertonic. Hypertonic means relatively less water, relatively more solute. So maybe this is 10% solute, and 90% water. Okay, more water, less water, hypotonic, hypertonic, and water always flows from hypotonic to hypertonic. That's the thing to memorize. Water flows from hypotonic to hypertonic. I want to emphasize that the interactive nature of this is really key. So as I talk and as I say something, I want you to chat in. That's what tonight's show is all about. Okay, it's all about your questions. So Gabriella and Tishal and Larilyn, keep on asking questions. That's what I really appreciate. So there's a device that's called a YouTube. They tend to make versions of this on AP tests. And, you know, the tough thing about a semester one final review is that your teacher's going to throw in questions that won't be exactly AP bio exam questions. They might be more questions that your teacher uses on their test. They might be a little bit more content focused and a little bit less conceptually focused. And you know, the only way to deal with that is learn the content. So here's YouTube. It's got one of these selectively permeable membranes over here. You can actually screw this thing apart. Like this actually really exists. The membrane, again, in this case, it's permeable to water, but not to the solute. We're pouring sugar in, sugar, sucrose, disaccharide. So what's going to happen? Water will move from this side of the YouTube to this side of the YouTube. That movement is osmotic pressure. And Kebab, you're killing me. You're killing me. Because I really just wanted to do units 1 and 2 tonight, but break down cellular respiration and Calvin cycle and the Krebs cycle. That would take a long time. Kebab, hang on, and maybe we'll get to that at the end, okay? But I really wanted to sort of like do part tonight and part on Thursday night. And I'm seeing a question from Laura. Do hypertonic and hypertonic refer to solute concentration? Hypo equals less, hyper equals more. So no. Okay, Laura. I'm going to go back over here. Well, actually, this slide will do fine. This side is hypertonic to this side. Why is it hypertonic? Because hypertonic solutions have relatively more solute and relatively less water. The tonic is the thing that gets dissolved. All right. So hypertonic, a lot of tonic, a lot of the stuff that goes in. Hypotonic means less solute. So Laurelyn, you're thinking about it. You're just thinking about it in the opposite way. So you just got to flip it around. Hypotonic is less solute. Hypertonic is more solute. And okay, Laurelyn, good. I think we straightened that out. And Jax, we'll do that about phospholipid heads and tails as soon as we talk about osmosis. All right. And I appreciate other people chiming in with answers too. That's awesome. Okay, so what's happening on a molecular level? You know, water forms hydrogen bonds with things like sugar molecules. So basically what happens is that water is going to go in, it's going to interact with these solute molecules and essentially get stuck on the hypertonic side. You know, water doesn't know anything. Water's just randomly going back and forth. But when it gets to the hypertonic side where the solute is, it interacts with it and stays there. And that's basically how the system works. So gummy bear places a student places a gummy bear in a bowl of water overnight. The next day, the gummy bear has expanded. Explain. Okay, so explanation. The gummy bear is what compared to the water? Can anybody answer that? The gummy bear is what compared to the water? I'd like a word to go into the chat. The gummy bear is what compared to the water? And you're choosing between hypotonic and hypertonic. What is it? I know there's a little bit of a delay in Zoom. And Gabriella, fantastic. The gummy bear is hypertonic to the water in this bowl. And so what happens is the water in the bowl flows into the gummy bear and the gummy bear expands. That's why it looks like this at the start of this fun experiment that you can do at home. And it looks like this at the end. Let's see what I said here. Gummy bear is mostly sugar. Water in the bowl is 100% water. Water flows from the hypotonic solution in the bowl to the hypertonic gummy bear. A really simple application. And here's something that isn't working. What a bummer. We're not going to do that. So let's go on to the next slide. Okay, so osmosis in plants. Use the principles of osmosis to explain each of the images below. If you haven't already had this image in a test, you will, because teachers love using images like this. Okay, let's try this again. The environment is what compared to the cell? Is the environment hypotonic or hypertonic? We're looking at this third one over here. I don't know why I started that way, but the environment is what compared to the cell? Let me see it. Let me see it. Let me see it. I saw some answers, and can I see some answers now? Okay, now, okay. The environment is hypotonic. Hypotonic. I'm seeing a lot of that. You guys are fantastic. You're killing this. You're going to do great on your semester finals if your knowledge on other topics equals your knowledge here. So basically, here we go. The water is going to flow from hypotonic to hypertonic. Hypotonic to hypertonic. So a plant cell that's placed in a hypotonic solution will become swollen, because water will flow into it, and the flow of water will be constrained by the cell wall. In this first situation, some of you anticipated me, water flows from the cell into the environment. That means if water is always going to flow from hypotonic to hypertonic, that means the environment needed to be hypertonic. And in the middle, that was isotonic, and the text is just going to repeat what I just taught about. So I think we're doing okay. Let's go on. Okay. Elodea in its normal freshwater environment. Elodea is a little water plant that you can find in lakes and streams and stuff like that. And here's what happens when Elodea is placed in salt water. That's because in this question on the right, the cells are what compared to the salty environment? In this diagram, what are the cells compared to their environment? I want to see a word like hypotonic or hypotonic. What do you think's going on? We've placed the cells in, the cells. I'm not sure if I'm getting the right answer here. Okay, so basically here, what you see is this. You see how the membrane is sort of shrunk away from the cell wall? You can basically see it in all of these. Well, that's because water flowed out of the cells. Water flowed out of the cells because when you added salt, that made the environment hypertonic to the cells. The cells were hypotonic to the environment. So water leaves the cells and the cells shrivel up. Just want to say I've got a song that's called Osmosis. I'm not going to play it now. You can find it on the Science Music Videos channel, and it explains all of this in a rhymed form. It's super fun. The salt makes the cells hypotonic to their environment. Water leaves the cell, the membrane peels away. Your teacher might or might not have you know this term, but it's called plasmolysis. Tishao is saying the cells are hypotonic. Breyer is saying if it's hypotonic, wouldn't it be full of water? The cells were hypotonic to the environment, and basically what the water is doing is it's leaving the cells to try and equalize the osmotic concentration on each side. So water flows from hypotonic to hypertonic. Jax is getting it 100% right. So water leaves the hypotonic cells to flow into the hypertonic salty water that's placed in the environment. And that's why the cells are shriveling up. And again, I hope you had the opportunity to do this in lab. Osmosis in animal cells, I think at this point we might be overdoing it, but I'm just going to jump right to the answers. In the left side, the cell is hypotonic to its environment. So water leaves the cells and the cells shrivel up. In the right side, here's a term we haven't used so far. There's an equilibrium of water flowing in and out of the cells. So the cells are all staying the same size. That makes the environment isotonic to the cells. And on the right side, the cells were hypertonic to their hypotonic environment. So water flowed into the cells. And because these are animal cells that just have a membrane and they don't have a wall, the cells burst. And in fact, for people who work with cells in culture, they need to use a slightly salty solution to grow cells so that they don't either shrivel up or burst. And that's what it's all about. That's what I just said. Okay, folks, how are we doing? Okay. I'm not seeing anything in the comments, but people are responding to my questions, so I'm pretty happy with how things are going. So there's a protist. It's called a paramecia. I really hope you got to see it in lab. You can just see it under a microscope. And basically, they have this organelle. It's called a contractile vacuole. And a contractile vacuole basically contracts to squirt water out of the paramecium. Why? Because protists, with their cytoplasm, protists are single-celled eukaryotes. Water flows in to the paramecium. Well, it can't expand indefinitely. And basically what happens is that it uses energy to pump water out through this organelle that's called a contractile vacuole. Water moves into the cells by osmosis, and the contractile vacuole contracts to get it out. I think there's going to be a problem with the next slide, but if you can sort of imagine this thing contracting, I'm sorry, I can't show it to you. It contracts and that forces the water out. This is what the contractile vacuole looks like. And hey, let's see. Look at that. Okay, that's the contractile vacuole forcing water out of the cell. Why? Because the cell is hypertonic to its watery environment. Water flows into the cell. The contractile vacuole forces it out. That was great. Okay, so this is actually a question. This is taken from a question that was in a past AP Bio exam. You should be able to read and understand what's going on here. So here's the contraction rate of a paramecia's contractile vacuole. Here's the molarity of the solution. Notice the relationship. As the molarity goes up, the contraction rate goes down. If the environment becomes more hypertonic, then the cell can adapt by decreasing its rate of contractile vacuole contraction. And it can do the reverse in more hypotonic environments. So basically, when the paramecium is in pure water, its contractile vacuole has to work the hardest because that is when the most water will be flowing into the cell. It's when the paramecium cytoplasm is relatively the most hypertonic. But as the environment becomes more hypertonic, then the contractile vacuole doesn't have to work as hard. I'm seeing a question from Gabriella. So the prefixes of hypo and hyper, low and high, refer to the solute concentration and not to the water concentration. I'm going to run back a couple of slides just to show this definition again. Okay. Okay, so you'll be able to see this on replay, but I'm going to do something I would never have done to my students in real time. Take out your phone, take a picture of this slide, because basically hypotonic means this: relatively more water. Water's the solvent, okay? Relatively less solute. Hypertonic means relatively less water, relatively more solute. And I want to emphasize, these are always terms in relationship to one another. All right. There's nothing that's absolutely hypertonic or absolutely hypotonic. So if something is hypertonic relative to a hypotonic solution that's next to it, across the membrane from it, stuff like that. Okay. So that's I hope, sufficient for hypotonic versus hypertonic. Let's talk about water potential. Skipping this. Skip this. Oops, that was a bunch of marketing stuff. Oh. I'm going to skip something. That's terrible. Okay, so here's what I'm going to do. I'm going to go out to learn-biology.com. No, you won't be able to see that. I'm going to go to, sorry about this. Okay, so this is my website, learn-biology.com, and let's go here to courses, AP Bio. Let's go to Unit 2. Let's go to osmosis and water potential. Okay, I'm just looking for a good diagram that I can talk about this with. Okay. So embarrassing. Guys, just be patient, please. Okay. Yay. Okay. So, this is a superpower that I have. Oh, you're not seeing what I'm seeing. That's bad. Okay. Hmm. All right. Go back here. Okay. So, um, water potential. Water potential is just a mathematical way of dealing with osmosis, and you can put it into an equation. And so we've already talked about this. You pour sugar into the left side, water will rise on the left side of the tube. Okay. Okay. Oh, you're really watching me flail here. Okay, I think I got this. Um, I'm trying to find a good slide about water potential for you, and I can get it, but it's just going to take me a second. Okay, sorry about this. Beauty of live TV. So here we are. Okay, fantastic. Sorry about that. All right. So, water potential. Thank you for not leaving. Water potential is represented by this Greek letter, which is called psi, kind of weird. And basically, water potential is the tendency of a measurement of water's tendency to move from where it is to where it's not. So, adding solute, and this gets really, really kind of confusing, adding solute decreases water potential, right? Whereas adding pressure increases water potential because it's about water moving from where it is to where it's not. Okay. So if you add solute over here, that decreases the water potential on this side of the YouTube. Water will move from here to over here, from higher water potential to lower water potential. Why is it lower? Because you added solute. If you press on a syringe, water will go out of the syringe. Why? Because you've increased the water potential in the syringe and water will flow away. So, what is water potential? Just memorize this rule. Water flows from areas of higher water potential to areas of lower water potential. And the formula that you have to know, there's a more complex version of this, but in AP Bio, all you have to know is this: water potential equals solute potential plus pressure potential. Here's what everything represents. And remember, adding solute lowers water potential. Adding pressure, like pressing on the plunger of a syringe, increases water potential. So here we have this put into um, numerical form. Pure water in an open container, the water potential is 0. It's measured in units that are called megapascals. That's something about pressure. It's from physics, and that's it. Rocket Launcher is asking, will there be an equation sheet on the AP Bio exam? There absolutely is. You don't have to remember any formulas. And I'm seeing a couple of people confirm that. So if you add solute over here, you've lowered the water potential, all right? Because now psi sub s, solute potential, is a negative quantity, negative 0.23. There's no pressure on either side. So on this side of the U-tube, the total water potential is negative 0.23. On this side, it's 0. Water moves from higher water potential to lower water potential. Water moves from this side of the U-tube to the other side of the U-tube. Okay, so that is one example of water potential that you probably had to deal with in your biology lab. If you did the potato core lab, you can also explain what happens in the potato core lab based on water potential. So basically, here's the deal. You put potato tissue into water. The cell has a lower water potential than the water in the cup. Why? Because cytoplasm has all these solutes in it, and solutes lower water potential. So, what'll happen over time? Water will flow from the cup where there's higher water potential, it's 0 megapascals, into the cell, which is -1, and that'll continue until the system equalizes. And that's because the pressure generated by water flowing into the cell will eventually equal the pressure pushing back by the cell wall. So eventually you'll reach an equilibrium and the potato cells will stop expanding. That's what happened in the potato core lab if you did that. They don't expand indefinitely, they only expand until the pressure of water flowing in equals the pressure generated inside the cell by the cell wall. Um, Okay. And then I'm seeing a question from Jax. Is it low water potential because there are less available water molecules? Basically, meaning that they're more bonded to the solute. That is a good molecular explanation. I don't think you really need to worry about it at that level. You just need to know water always flows from higher water potential to lower water potential, and that solute decreases water potential and pressure increases water potential. I think that might be the end of my water potential explanation, which really took a long time for me to get started. My apologies for that. Here's a cool thing. See a crack on the sidewalk like this, tree roots that have pushed up the sidewalk. You know what that's about? It's about osmosis. Tree roots don't have muscles or anything like that, but the cells expand through osmosis. Every time it rains, water flows into the roots, and the roots grow, and the cells grow by actually expanding by pushing against their wall, and the roots expand by osmosis. And that pressure, it's actually called osmotic pressure, is enough to crack concrete. And you see that on every sidewalk in the United States of America and any other country as well. So that's it. Okay, so I think what we're going to do now is we're going to go back here and we're going to go back here, and that's okay. Good. And so now we're going to go back here, and there were some questions that came up before. I'm going to look through the chat because I'm forgetting what they are, but if you put them into the chat again, that'll help me. So Anybody else got a question? Oh, okay. Jax had a question about uh, cell membrane structure. Okay, so what we're going to do is we're going to do organelle structure. Excuse me, what we're going to do is we're going to do about membrane structure, and then we will go to talk about like, we won't do them all because we can't do them all, but we can do like smooth versus rough ER. It's basically what we can do. So let me go like this. Let me go like this. And let's find phospholipids. It's the best way to do that. I've got it over there, but I think I have it in a better structure, better presentation a little bit further on down. That's all the stuff about membranes. Okay. Okay, so little moment. This is a phospholipid. Phospholipids are the basic structural components of cell membranes. The thing to know about them is that they have a tail that's hydrophobic. These two fatty acid tails, they don't like interacting with water. Why is that? Because they don't have any functional groups that interact with water at all. So these are hydrophobic, water fearing. Also, you might call them non-polar. All right. So non-polar side, hydrophobic side. This is a molecule called glycerol. It plays a structural role of holding the whole thing together. And this is the head of the phospholipid. And the head of the phospholipid has a phosphate group over here. And it's got another piece over here. But we're mostly going to focus on the phosphate group because you see that negative charge? That makes this phosphate group interact with water, form hydrogen bonds with water. That makes the head hydrophilic. The head can also be described as polar. So we've got a polar hydrophobic, polar hydrophilic head and a non-polar hydrophobic tail. This is a cartoon representation. So remember, this is the head. It's polar and hydrophilic. This is the tail. It's non-polar hydrophobic. If you put these into water, then these phospholipids will spontaneously organize themselves like this. They don't have to, there's no enzyme, there's no chaperone protein or anything like that. This is the lowest energy state for these molecules. And if that just went like, whew, just don't worry about it. The basic idea is that you mix phospholipids with water, they'll form a bilayer. And they'll form bilayer that'll create one of a couple of structures, but one of them is a sphere. And that sphere has phospholipids on the surface facing out towards the watery exterior, and then on the interior as well. And if you can imagine this as actually something spherical, you've basically got the framework of a cell membrane. You've got a little spherical bubble with a phospholipid bilayer. So that's the basic idea. In a solution, the heads bond with water while the tails form a water-free zone, and that creates a bilayer, and that's the framework of the membrane. And the phospholipid bilayer is further stabilized by some weak bonds that happen between these fatty acid tails over here. And I've got a really cool animation of this that I'll show you. It's from my song Membranes. I hope the sound goes through.
Because when phospholipids into water get submerged, a phospholipid bilayer structure will emerge. The tails hang together in a water-free zone. Hear the hydrophobic mode, water leave me alone. While the heads are sticking out, touching all those H2Os, tails in, heads out. It's how every membrane goes, tails in, heads out in a cellular sphere. It's the bilayer basis of membranes everywhere.
All right, enough of that. But that's in my membrane song. You can watch that again. The basic idea is that phospholipids will spontaneously form themselves into a bilayer structure. Jax, I hope that did it for you. I'm seeing a comment by Gabriella. Functional groups are hard to remember because there's so many. How much do we have to know of the common ones? You kind of need to know them in context. So let me talk about the rough and smooth ER. That was Breyer's question. And then Gabriella will talk a little bit about functional groups. So let's go like this. And let's go like this. And let's talk about the smooth and the rough ER. Where is that? That's going to be in all this stuff about. Okay, here we are. Fantastic. ER, smooth ER. Okay, beautiful. Great. Here we are. Okay, so let's just roll back a little tiny bit. Let's talk about the endoplasmic reticulum. All right. So the endoplasmic reticulum is this and this. It's numbers 2 and 3 in this diagram over here. So this is the nucleus over here at 1. Here's the nuclear membrane. And then we have the rough ER and the smooth ER. What is it? It's an interconnected series of channels between the nuclear membrane and the Golgi body. And there's a rough ER and a smooth ER. The rough ER is rough because it's studded with ribosomes. And what it does is it synthesizes proteins that are associated with phospholipids. Either they'll be included in a lysosome, they'll be sent to another organelle, they'll be sent to the membrane, or they'll be exported. And so I have a picture over here of what happens to white blood cells when they're creating antibodies. That's a kind of protein that's fighting off an infection. They get filled with rough ER. Why is that? Because that's where they're synthesizing these proteins, antibodies that fight off viruses and bacteria and stuff like that. I could explain how the ribosomes get to the rough ER, but I'm not going to do that right now just because there's only so much time. And T Shell is saying, isn't the nuclear membrane contiguous with it? Yeah. And I show sort of like a space between the two. You know, that's an interesting question. I actually don't know the answer to that. I don't know if they're just really close to one another or if, like, for example, there'd be like a little continuity over there. There has to be a way for mRNA to get directly into the cytoplasm and not go into the ER. Sometimes it goes into the ER and sometimes it doesn't. This whole thing is called protein targeting, and I have a tutorial about this on my website. Smooth ER. Smooth ER is on the outer side of the ER network, and it lacks ribosomes, but it has many enzymes, so it's doing a lot of biochemical stuff, and its functions really vary by where it is. So it can be synthesis of lipids, converting toxins into soluble forms that get just excreted from the body, carbohydrate breakdown and synthesis. Those are things that the smooth ER does. But for an AP bio class, at least the way that I taught it, if you just know that the smooth ER doesn't have ribosomes and the rough ER does, and then some stuff that I talked about before, rough ER is making proteins for inclusion in lysosomes, organelles, or for export for the cell, you are fine with this. And Eddie's asking whether the video saves. Yeah, the video is going to be available for watching on YouTube. And just so that you know, if you go into the Science Music Videos channel, I've got review videos about all of this stuff. That's going to be really useful. Gabriella had a question about functional groups. And I just want to say a couple of things about it because it's not impossible that you'll get a functional group kind of question. So this is going to be way back here. Okay. So functional groups tend to kill AP Bio students because it involves a lot of chemistry that often you don't have access to. So what do you really have to know? I have a tutorial about functional groups on learn-biology.com. You mostly need to know them in context. So I'm going to show you a couple of examples. So like over here, you need to know that like if a molecule like this triglyceride, it's a fat molecule, if it doesn't have any functional groups, like this one does. I mean, it sort of does insofar as it sort of has these methyl groups over here. It's going to be it's going to be nonpolar or hydrophobic. It won't dissolve in water. But when you add most of the functional groups, like this is a hydroxyl group over here, and you don't even need to know its name, but you just need to think like, oh, it's an O and an H together. Oh, that's kind of like a water molecule. Like if there was another H, that would be H2O. So this makes it polar. So this is why sugar dissolves in water, because it's got all of these OHs, also known as hydroxyl groups. So one thing to know is that there are certain functional groups that make molecules polar or hydrophilic. It makes them able to dissolve. Um, Phosphate groups over here, they're in the head of a phospholipid. That makes sense. Phospholipids, that's why the phospholipid bilayer forms. Functional groups are super important in terms of protein structure. So, like, for example, here's a hydrogen bond that's forming in a protein, determining the part of the tertiary structure. Here's a sulfhydryl group. So I don't know what to say, because it's probably not really a great idea for you to spend a lot of time memorizing all the functional groups right before your test. You have a lot of things to learn, but at the same time, I always taught them to my students because it made it really easy to talk about a lot of biology. Let's see what else I have here. That's it. So, Gabriella, that's not the most complete answer. If you want to learn more about functional groups, you can do it on learn-biology.com. But I think it's mostly about knowing the functional groups in context. All right. Seeing a couple of new comments come in. T-Shell is also asking for Krebs and Calvin, and really, let's see. If there are no more questions, because we've only got, I was only planning on being here for about an hour tonight. So anybody else got something from units 1 and 2? Okay. Larilyn, that's a great question for units 1 and 2. So let's go like this. And let's talk about membrane transport. Okay. Okay, membrane transport. So the main thing to know is that diffusion is how many substances enter and leave cells. We've already talked about osmosis. So diffusion is tendency of molecules to spread out from where they're more concentrated to less concentrated. You drop a little bit of ink into a jar of water, it spreads through diffusion. Molecules spontaneously flow down their concentration gradients. And that's an important term to know. That might show up on an FRQ. Things are going to move down their concentration gradient. So here's oxygen flowing down its concentration gradient from higher to lower concentration. Things will flow across membranes down their concentration gradient. Diffusion is also known, I sort of garbled that, diffusion is also known as passive transport. So here we have molecules that are diffusing from higher concentration to lower concentration. Again, higher concentration to lower concentration. No cellular energy is required. The energy is already present in the kinetic energy of the molecules. There are two forms. Simple diffusion is when things flow right through the phospholipid bilayer. Facilitated diffusion is when they require a protein channel. To facilitate something is to make something more easy. And if you speak Spanish, fácil is easy in Spanish. It's also in French and Italian too, I think. So facilitated diffusion is diffusion that's been made easier. It's been made easier by this protein channel. Now, why do some things enter cells by simple diffusion and why do others require facilitated diffusion? Simple diffusion is for small non-polar molecules like oxygen and carbon dioxide, and also for non-polar molecules, so lipids like steroids and fats, they can use simple diffusion to, excuse me, make their way right through the phospholipid bilayer. So simple diffusion, no protein required. Things flow down their concentration gradient. Small nonpolar molecules or lipids are able to do that. Okay. I saw a comment about um, aquaporins. Aquaporins facilitate the flow of water across the membrane because water is polar and it doesn't diffuse that easily across the phospholipid bilayer. So aquaporins are proteins that actually get sent to the membrane to help water diffuse through. Facilitated diffusion is for polar and ionic substances. So what's an example of a polar molecule? Glucose. Polar. It has all those OHs on the outside. Amino acids. They also, they might have acidic parts to them or basic parts, obviously. They need proteins to help them diffuse through. And then any ionic substance, anything that has a full charge, that is also going to need a protein channel. So that's basically what I just said. Now, just to finish this discussion, the opposite of diffusion is active transport. And active transport requires a kind of pump to move things from lower concentration to higher concentration. Things get pumped against the flow of diffusion, they get pumped up a concentration gradient. And that's what active transport is all about. Pumping a molecule up its concentration gradient. It requires energy on the part of the cell. That's usually ATP, which gets broken down to ADP and phosphate. That's powering that process. But it can be electron flow. And on Thursday night, when we talk about the electron transport chain, for example, it's electron flow that powers proton pumps that make the synthesis of ATP possible. All right, so membrane transport, good. Laralyn, that did that. So, folks, more questions. More questions. I'll go back to our list of topics. Let's see if I can easily find that. Just while I got y'all, got everybody's attention. Remember, learn-biology.com, tutorials about everything. Download my checklist. And remember, if you really want a long, detailed, like 3-hour version of this, I have one. You have to pay for it. And it's going to be on Zoom. It's going to be on Saturday morning, December 7th. And if you go to apbiosuccess.com/SEM1review, you can find out about registration for that webinar. Here were the questions. Okay. So I think we have just time for like 2 more questions. Okay, phagocytosis and pinocytosis. Okay, so let's talk about that. All right, so we just talked about simple diffusion. Actually, let me find the slide first. Oh, that's funny. We were right there. All right, so there's the flow of molecules. That's passive transport, that's active transport. And we talked about simple diffusion and facilitated diffusion. But then there's what's called bulk transport, where things get moved in bulk. Lots of stuff gets moved. So bulk transport usually involves vesicles, big movements of the membrane. And Gabriella, you're mentioning phagocytosis and pinocytosis. That is going to be up to your teacher. That wouldn't be on the AP Bio exam, but you would have to know about the difference between the 2 terms I have here on the slide, which are endocytosis and exocytosis. So here's what endocytosis is. The membrane pinches in to surround either a molecule or a particle or a cell. This says large particle, but this really looks like a bacterial cell. So the membrane is sort of buckling and surrounding this, taking this in, and that is creating a cavity that becomes a vesicle. So that's endocytosis. Endo kind of sounds like into. That's how I helped my students remember it. So endocytosis means into the cell, right? That was kind of silly the way I did that. Um, Phagocytosis, here I have a slide that shows it. So this is a kind of cell. It could be an amoeba, and it's actually swallowing up a bacteria by extending these extensions of its membrane, surrounding it. It makes it into a vesicle, and then that vesicle fuses with a lysosome, digesting it. So that's phagocytosis. It's sort of endocytosis on the biggest scale. Um, there's a really cool thing that I can't resist teaching you about. There's this thing called receptor mediated endocytosis. And that's where the endocytosis doesn't happen until some molecule binds with a receptor, and that causes the membrane to buckle in. Well, remember when school was shut down because of SARS-CoV-2 and we all had to wear masks and things like that? Well, that happened because the SARS virus learned how to bind with a enzyme that's on the surface of some of our cells, epithelial cells. It's called the ACE2 receptor. And when it bound, here's SARS-CoV-2, when it bound with that receptor, that tricked the cell into basically taking the virus into the cell. And then the virus continued its replicative cycle. And that was really what the pandemic was all about. HIV does the same thing. It binds with a different receptor. Exocytosis, I cut off the Y over here, so it's exocytosis. That's where cells put things into vesicles. Those vesicles start in the rough ER, they go to the smooth ER, they go to the Golgi, and then the Golgi buds off vesicles. And then those vesicles fuse with the membrane and they dump out their contents. Exo kind of sounds like export, and that's what happens in exocytosis. So here we have a B cell that's secreting antibodies through exocytosis. Vesicles fuse with the membrane, releasing their contents. That's what you need to know. So anyhow, Gabriella, really just endocytosis and exocytosis. I just want to say this because we're getting really close to our end time. Just want to emphasize one more time. Hope you join me on Saturday. You get 3 hours of this. It'll be fun and you'll learn a lot and it'll set you up for really a lot of success. I will go over units 3 and 4 on Thursday. So I'm sorry for those of you, but I couldn't do everything tonight. So Thursday, I'll go over units 3 and 4. I'll try and throw in a couple of FRQs. Here's the link to sign up for this webinar, apbiosuccess.com/sem1review. Tonight, if you've got a final tomorrow, go to learn-biology.com. You can sign up for a free trial. And once you're in learn-biology.com, you can get quizzes on every topic in AP Bio. And the quizzes are designed to help you learn. So the quizzes give you feedback, they give you hints. And if you do the quizzes, you will wind up learning a lot. I've also got some games on the site, flashcards. It's really designed to optimize biology learning, and it's all about AP Bio. It's specifically an AP Bio curriculum. And download this checklist. It's really going to help you in terms of telling you exactly what you need to study, what you need to get your head around. Folks, I wish I could stay here all night. That would have been really fun, but we're going to bring things to a close since it's 6:59. I am very, very grateful to you for spending time with me tonight. This has been really, really awesome. I hope I see you again Thursday. I hope I see you on Saturday as well. So everybody be well, stay safe. Mr. W signing off. Good luck in learning biology, the best subject in the world. I'll see you folks again soon. Everybody be well.